So this is the final lecture, or rather an interactive session for the course, Introduction to Professional Scientific Communication. So today we are going to have a discussion as to how effectively you can communicate. What is your research problem? You are doing a PhD or you are doing a postdoctoral fellowship, and someone is asking you what is that your research project is, and if that person is not from your domain, he may not be a scientist, he may belong to some other field. So still, you are expected to give an answer that is convincing, and therefore the other person can appreciate what you are doing. So this is also an effective way of communication. So we are going to use our volunteers here, students from our department and our lab, where I would simulate a situation wherein I am going to randomly ask questions as to what is their research project, and they are going to give an answer. And then we use that answer as our learning point, and we will discuss as to how that answer can be better, right. So that's the example, and these are simulated situation, not necessarily that everyone who is telling that giving the answer they are as bad or as good. I mean, this is kind of a simulated situation. Therefore, we can capture the different kind of people normally you come across when you ask questions on the research project and so on. So that's the focus of this particular session today. So let me introduce my participants here, volunteers here. 8 of them are here. So what I am going to do is I am going to ask them to explain exactly what they are doing in their research project. So that should be not more than 1 minute. It should be very short answer. At the same time, they should be able to tell what they are doing to someone who is not an expert in this field. So that's the prerequisite, right? So that's what the conditions are, and I would start with Akansha. Akansha, so can you explain exactly what is your research project, and in about in 1 minute time? You can hold it close to your okay. Just like this.
So hello everyone. So I am basically working on deciphering the role of glycogen synthase and autophagy. So it is long known that glucose, which is the major energy source in the body of an organism, is primarily converted to glycogen, and this glycogen is broken down to provide energy to the organism. So in our body, the neurons, which are considered to be very much active, metabolically active, despite the high energy requirement, they prefer not storing glycogen. However, they have the active glycogen synthesizing machinery within them. So my project mainly aims to decipher that why at all glycogen synthase, although it is present in the neurons, what is its role in the neurons. And the second part of my work mainly deals with deciphering the role of glycogen synthase and cell cycle. So people have found that glycogen, it is the amount of glycogen, it varies when a cell progresses in the cell cycle. So my project also aims to establish that if neurons are keeping glycogen synthase inactive, so is it so that activating glycogen synthase can trigger the entry of a neuron into its cell cycle, thus leading to its death.
Right. Very good. Right. So we'll go to our second participant. I'll hand out to Smriti. Can you, in about a minute, explain what is your research project?
Hello everyone. I am working on the model sporadic Alzheimer's disease, and which has a metabolic basis of its etiology. So basically I am looking over the insulin signaling pathway, and what is the scenario if we look at glial and neuronal crosstalk. So basically I am finding the overview and some candidate genes involved in the pathway, and the crosstalk between glia and neurons using a co-culture system. And I'm working on animal model, which is surgically induced model, or of sporadic Alzheimer's disease. So, taking these two models, I am just looking on the insulin signaling pathway, that what are the main genes which are mainly compromised, and and what is the status of glia and neurons, and in the way I'm trying finding out that who are the real culprit for this disease pathology, they are glia or neurons.
Good. So, we have had two such examples now. So, we basically requested the student to present in about a minute as to what exactly the research project he or she handling for the PhD program. So, that is the question, and you have witness to that answer. So, before we go and look at the aspects as to how you would or how you should present your project to someone who is not expert in the field, let us look into some of the thumb rules. So, that is what is shown here in the slide. The question is how to explain your research project to an outsider, meaning somebody who is not working in your field. So, these are some of the issues that we have discussed at length in the course, that whenever you build something new, you are going to say something to somebody who has not been exposed to that topic, or you are not yet introduced as to what you are doing, you have to keep two important points in your mind. One, given and new. So, this is something that we have discussed, whether it is written communication or it is oral communication, this is a very important element. So, you have to always start with something that the other person is aware of, he knows about it or she knows about it, and then you build and explain as to what possibly they may not know. So, this is the fundamental kind of a structure of your narrative. It should be given and new. Given is something already known to the audience, you start there and then move to something new. The second element is you have to convey whatever you are doing in the most simplest form possible to begin with, and then you build the complexity. Otherwise, the person who is an audience or somebody is asking question or interested in what you are doing, he or she may not be able to follow what you are doing. So, you have to always start with something that you as well as the audience or somebody who is talking to you, is a common point that they know, and then you go to something that probably they may not know because you are the expert, not the other person, and then emphasize and tell what you are doing and make it simple first and then build complexity. So, if you do not do that, then probably you are not communicating effectively as to what you are doing. So, the very purpose of the communication is that you are trying to tell the other person what you are doing. So, if you cannot make the other person understand and appreciate what you are doing, then you failed as a scientist in communicating what you are doing, right. These are two important elements, and then this is with regard to any section, whether it is an introduction or you are talking about a method that you are using, or what would be the conclusion or what would be the impact of your finding. In every section you have to follow this given, new, simple, and then make it heavy, what you call as light and heavy. Beyond that, there are several elements in your narrative, like we discussed abstract. So, as and when somebody asking you as to what you have done or what are you doing for your research, what you should explain is something what you would write as an abstract in a research paper. So, it is about say 200 words, probably in 1.5 minutes or 2 minutes you will be able to tell that, right. So, how heavy or how simple it should be depends on the other person. So, this is a point again we have emphasized when we say, when we discussed on the points on oral communication as to you should know about your audience, right. You should know who is the other person. So, how much probably they know, and therefore, you should pitch it to that level. The first thing is that you should use this principle given and new, light and heavy for each one of the other bullets that are listed. Provide a brief introduction to the broad topic. So, you cannot start with something so specific and expect other person to know what you are doing. It has to be very broad, aspect, and then define the research question. So you have to say, give an introduction and ask the question as to, this is the question that I am addressing and why that question is important. And to ask this question, you are going to approach the problem. So you always, in research, in basic research, you always have an hypothesis most often. So you have an hypothesis, you have to state, and then how are you going to test the hypothesis, and if you already have got results, you say what is your observation, and finally you conclude as to what do they mean possibly, right. So this is the basic structure with which you should be able to convey what you are doing, right. So that's very, very important. Now give an example, I'll give an example as to why this is important. If somebody is asking me, where do you live? So if I say 510, which is a fact, I live, my, the residence, the number is 510. So that's true, but it does not convey anything to anybody, especially if the person who is listening to this, my answer, is somebody who is not in this city, who is not in this state. So 510, although it is the answer, it does not make any sense. So you normally start with something which is common point, right. If somebody is there in Kanpur, the city that I am currently living in, and I meet someone in Kanpur city and somebody is asking you, where do you live, then I should be telling, this smaller locality within Kanpur, there are a number of divisions, right. So I can say, for example, I live in the IIT campus. So everyone in Kanpur city know where is IIT is located. But if somebody is there in Delhi and is asking me, where do you live, I cannot give the same answer that I live in IIT campus. Then they will be thinking I am living in IIT Delhi campus, right. That is not the answer. So you should say, Kanpur is my city where I live. And if it is somebody who is outside the state, so I should say, I live in UP, and then I could say which city it is. So it depends on to whom you are giving the answer. So you should always find, the common point, what you call as given, so that they also know, and then you try explaining it. So that is basically is what given in the address. If you write anywhere, any letter to anyone, there is an address, postal address. It always starts with, the state, the city, and then within the city there are different, sub pockets that you call Indranagar or whatever, right. And then you have a street, in that there may be an apartment, and then you have a flat, and that's how the number is, right. That's how the address is given. So that is exactly the way you should be answering to anyone who is asking you a question, right. That you should always remember. So if you have used internet and somebody wanted to search as to where is a given particular a shop or whatever is located using Google map, if it zoom in exactly to the street and then show where is the shop is, then I cannot make out anything. So what I should do? I should do the unzooming, make it such a way that now I can see, for example, entire city, and then I can relate these are the different section of the city that I know, these are the locality. I live in this place, therefore the shop that I am looking at is somewhere, for example, on the northwest of my current locality. So you are able to relate it to it. So you should know where you are and then relate to where is other location is. If you straight away zoom there, then you cannot make out anything as to where it is. So these are some of the examples I give to drive home the point as to how you should be presenting yourself as to what research project that you carry out, right. So with this little introduction, I go back to the same two participants. Now, for example, Akansha, right. In less than a minute, using these principles, now you can redo your introduction, right. So you always keep this in mind, given new. So you should start with something that is the other person should be aware of, and then connect it to something new, should be light, simpler, and then should bring complexity, and there should be a broad overview, question, hypothesis, if you have results, and then what could be the impact. So, this is the structural features of any answer. If you could now do it again.
So we all know that the food we eat is mostly to obtain energy and glucose from it, and this glucose is converted to glycogen, and whenever our body does not have whenever food is not available, then this glycogen is broken down to provide energy to us. In our body there are several different organs, one such is the nervous system. So the nervous system, the neurons, they do not store glycogen. We know that they are metabolically very active. We have to, they are always in function, but still they do not store glycogen. So my project aims to question that why at all neurons do not store glycogen, although their metabolic requirement is so high. So my project aims to decipher that. Now the second part is that
It's fine. So, let's look in that. So as you can understand, it's not easy. It's very easy to tell that this person or that person did not give a good answer. It's very easy to tell that this person or that person did not give a good answer. It's not satisfactory. It's not convincing. It's very easy. But if you are the person to give that answer, you will find it difficult, right? That's the fact of the matter, right? These are all just to simulate that kind of a situation. Therefore, you can appreciate the difference, right? So, always you have to start with something given, right? That, in a revised narrative, Akansha did beautifully. That is, that all our tissues have most of the tissues except neuron have the ability to store glycogen. So, you eat and carbohydrate in your food is broken down, converted to glucose, transported via blood, and then the tissue which has got cells store them as glycogen for their energy needs. But one exception is the neurons. They do not store glycogen. They use probably glucose, but they do not store glycogen. But what is interesting is, though they do not store glycogen normally, they do have the machinery required for glycogen synthesis. One of the main enzyme, what you call as glycogen synthase, which helps in the synthesis, in the formation of glycogen. So if they don't store glycogen, why should they have glycogen synthase? That's the question that she is addressing, right? So in the narrative, she did well, and she is able to give what is called as given, and she is able to introduce what is called as new. And of course, we can go on explaining as to what is her hypothesis, how she is going to address them, what would be the impact of her finding. So to help her, I am just putting an hypothesis. The hypothesis is that though the neurons don't synthesize glycogen, they do have this enzyme called glycogen synthase, maybe because it is very much required for the neurons to survive under certain conditions such as stress. Therefore, if that is true, if that is the hypothesis, my approach would be to provide stress to the neurons and then somehow block the glycogen synthase activity, and then now see whether neurons can survive, whether so that's the approach. And then if I can show or establish that glycogen synthase is indeed required for the neurons to survive stressful condition, the impact is enormous. I am going to say something altogether very different. So far, probably no one has really looked at glycogen synthase as something which can function to protect cells. And if this is indeed true, and if you could prove, then that is something new, novel discovery that you made, and that could have a greater impact, because you can use that understanding in protecting neurons, which is after all is the main problem in many of the neurodegenerative disorders. So that's the impact one can think of. So that's how you have to structure. So let's now go to Smriti. Now can you use this few guidelines and rephrase your answer?
Basically, I am working on investigating the uh, disease pathology of sporadic Alzheimer's disease, which is a most common form of dementia, and its etiology is having some metabolic basis. So broadly speaking, we have in our brain there are two type of cell populations. One is glia, other is neuron, and glia are known to be the guardians of neurons. But recently, some of the papers appeared which have reflected that in under stress condition, glia are actually not guarding the neurons. They are not protecting it. They are itself harming it in some or other way. So my project is using glial and neuronal crosstalk. I want to check whenever a stress condition arrives in particular disease condition like sporadic Alzheimer's disease, like metabolic insult in the living system. So glial cells are actually the guardians of neurons, or they are causing some harmful effects. So which are the real culprits of Alzheimer's disease? It's glia or neurons. So if we would be able to answer this question, there could be a better therapeutic implication for this particular disease condition.
Good. So this revised kind of version of the answer is able to bring out more points than what it did earlier. But still there are some points that one should keep in mind when you make any answers. One is that you should avoid as much as possible the technical words. So the technical words should be used only when you have no other option. So you should make it as simple. That's precisely one of the bullets. That is, you should be simple. If you can convey whatever you wish to convey using simple words, that's the best, because then certainly the other person is able to get everything that you want to convey, right? So you avoid as much as possible the technical words. You use them only when you have to, and if you have to, you have to define what it means. Otherwise, it does not convey anything. So you have to remember these essential points. Whenever someone is asking you a question, often they want to know what you are doing. They want to know what are your contributions. They are not asking this to test your knowledge. That happens only in exams. Therefore, it is not to, it is not the opportunity to, for you to show everything that what you know. So that you should keep in mind. So you should, make it as simple, avoid as much as possible the technical jargon, but convey what exactly you wish to do. And you do have to, at times go into the complex questions, but you have to build your case, right? So you have to convey in simple words. Therefore, the other person is able to get what you are trying to tell, and then move towards more complexity. That really helps. It's called the hand-holding. Like really, you have to help the audience to understand what you are trying to say, right? These are you can go on asking many such questions, and you keep improving. And you have to keep in mind that it doesn't come in one day or in one go. It takes years, so you have to practice it. One way to do that is to listen to others as to how do they answer. And you don't follow them, that means there is something that is not said in a way that should be told. Then you ask yourself question as to what is missing there. Then you will understand what are the bottlenecks or what are the elements that make it very difficult for you to follow, and therefore you can address them when you have an opportunity to explain to someone else and try to implement every time you do that. Then with practice it gets better. So communication, as I told you before, it's like a fine arts. More practice you do, more better you become, and exactly that's the case with scientific communication as well. So you have to, for you to write very good research article, you should have tested the waters for at least 10 times. You should have written at least 2 articles, tried your best, corrected by your supervisor or your teacher. Therefore, you know what issues that you had in your writing and how it can be improved and so on. So we looked at several other points that you should keep in mind when you do such uh, presentations, right? So these are some of the thumb points. Now we'll move to two more people. We have given a brief background. Priyanka, so
Hello everyone. So I am working on the project to decipher the role of nuclear protein 4, that is NOL4, during craniofacial development. So my interest on this project derived from my earlier interest in the project which I have worked on to understand genetic etiology of orofacial defect, that is cleft lip and palate, which is a multifactorial disorder, that is both genetic and environmental factor plays role in this etiology of this disorder. And also this is one of the most common birth defects where you find a gap in the lip and palate. So understanding a syndromic form of this cleft lip and palate, that is Van der Voort syndrome, which has lower lipids instead of additionally apart from cleft lip and palate as well. So understanding this genetic etiology of Van der Voort syndrome, I have earlier done next generation sequencing for whole genome in 5 affected members of the family and 7 unaffected members of the family. And applying these approaches, we have narrowed down to one gene, that is nuclear protein 4. There is a intronic mutation in this gene which causes the disorder in this particular family. So this gene is expressed during the development as well, and we have found that its expression in lip and palate during development using mouse and human embryo as well. So now I want to work on the characterization of NOL4 gene, like its role of during development in considering the both like face development and lip and palate development as well.
right. So this is another example, right? So let's look into how this narrative can be made better, right? So let's follow the same principle. That is, you have given, then introduce new. So you should never start with something new. That's the thumb rule. Never, ever start with something new. So I told somebody is asking me, where do you live? I cannot say 510. That's not the answer. I gave an example. And then it should be light, and you should make it complex if required, right? Not otherwise. So if you can rework on the project that Priyanka is looking at, we can start with in a very different way. I'll try to give it an alternate way of explaining what she is doing. The development, you must have seen the child is born, and then with time there are changes that happens in your body. Every event is controlled by gene, right? You have genes in your genome or chromosome that regulates as to how you develop. Same thing happens when the fertilized egg develops and forms the baby just before birth, right? So during this formation, there are a number of genes involved, and if there are some defect in the way the genes function, then some of the developmental events are not complete, and yet the baby is born. And one such condition is cleft lip. You must have seen few babies born with the lip not being fused together, and they may also often have the upper part of the mouth, which you call as the cranial. This is also part of the skull bone that is also not fused. You have a hole. Now this is a developmental condition. One can easily fix it by surgery, but however, we need to understand as to why such incomplete development happens. And that is exactly the project of Priyanka, where she tried to look at genes and their sequence in individuals that had this kind of deformity and compared it with the normal individuals who are otherwise having a normal, fully developed the mouth, lip and so on at the time of birth, and she could find one gene when there was a variation. Now the question is whether that is the gene that is causing that defect, and therefore she is trying to understand whether this gene does play a role in the in the development of the upper part of your mouth using model systems. She uses the mouse, she uses cell culture models trying to understand the exact function of the gene wherein she found a variation. So that's the project. So we can make it very, very simple to someone who is not exactly in the field by removing all the technical words, yet conveying what you are doing. If the person is asking, how did you look at the sequence variation? Then you can go and explain as to how did I do that. I can say there are several methods. One of the methods that I used is next generation sequencing. That's not upfront required. What is required is to convey that there was a sequence variation in a gene. That's more important than how you arrived at, right? So that's the way you have to build your responses, that you have to keep that in mind. Remember, all these are, we are giving different kinds of expressions to make you aware that there are often answers which are technically correct, procedurally correct, yet they may not convey what they wish to convey to everyone, right, unless you are in the field. And that's where we are trying to help, how to convey something that your project can, as an output that could come out, how it could help the society or in the knowledge base and so on. That's the example. So we'll go to the final one example. We'll go with the
Okay. As we in our daily lives also, we come across several environmental stress or in one such condition like fever or something, when we have we have higher temperature, whatever we regularly have in our body. So all this brings stress to our body, and our body is made up of cells and tissues. So it basically affects all the cells and tissues as well. So one such condition I'm trying to understand, when cells are subjected to stress. So how in normal condition, whenever the stress is there, then how cells are trying to cope up with that. So in general, whenever there is some stress, there are some specific machinery inside the cell, those who try to recover from that from such kind of conditions. So one such condition is metal stress, or any kind of chemical stress, or heat shock. Heat shock means that whatever our body temperature, 37, it's above to that, 40 or 42, it causes as a stress. And for this, in our in cells, there are stress granule formation, which helps to recover from the stressful conditions.
Fine. We will. So that's a nicely summarized work, but you can make it much simpler. So I'm going to narrate that and explain how we can make it simpler, right? So she spoke about stress, but we have not said what is stress. So that's what I mentioned some time back, that when you have any technical term, we need to define what it is and then say this definition is referred to as this. And then you start using that word, people would relate to what you mean by that. So always, when you are talking about something that the other person may not know, you should define what it is. Otherwise, the very first word that you use is something not given, it's new. So the person gives up that itself, right? So our cells are used to a particular body temperature. So our normal body temperature is 37 degrees. So all our biological activities, physiological activities are tuned for this particular temperature in all the mammalian species. So what happens to the cell when the temperature goes above 37 or goes below 37? Because that is not normal. So when the temperature increases above 37 or goes below 37, that's the condition that you call as stress. It is a stress because cells cannot function in that temperature. So that may be lethal to the cell because that can affect the cell survival. However, this shift in the temperature could be for a very short period, maybe within 10 minutes, maybe within 40 minutes, the temperature may come back to 37. Therefore, the cell should somehow survive that small period of higher temperature or lower temperature and wait for the normalcy to come back and then resume activity. And it happens all the time. As and when you have a fever, your body temperature goes up, and then cells experience much higher levels of the temperature, which certainly is not good, but still we all survive the fever because the cells have the ability to cope up with higher temperature. So how does it do, and what damages the temperature can cause? It can cause enormous, right? For example, the protein may not be folding properly. That itself is a huge stress, because abnormal protein can affect the way the cell functions. So one of the ways by which the cell reduces the load of abnormal protein is by arresting the synthesis of new protein. So when the cell is under higher temperature condition, what you call stress condition, it arrests the translation process, the process by which the cell makes protein. And that time all the mRNA which are actively translating, making the protein, are put into small regions, and they are aggregated, and certain proteins cage them together and not allowing them to make the protein. And such kind of foci, or small groups of such RNAs that are not translating are called as stress granules. And Rashmi's project is to understand how these stress granules, which are assembly of RNA that are not being translated, not allowed to translate, are regulated, and how they get back to initiating the translation when the cells come back to normal temperature. So this is a much simpler way of explaining, but you can make it much more simpler, right? So you can ask questions. You can say, what is your hypothesis? How I am going to address them? So you have to practice. The one way to practice is you type it out. Now you see how many technical words that you have. Try to remove them. Replace it with simple words. Replace them simple words. Revise for 10 times, 15 times, and then try to reduce words. Convey the same meaning but remove words. Then you will understand that you are using so many jargons, so many words, that not at all necessary if you want to convey what you wish to convey, right? So that's how you practice. You don't need to really orally communicate, but you can also start writing and try to remove, and finally arrive at a 3-4 sentence answer, which does not have any technical words, jargons, but yet you can effectively communicate. One final question on this to Rohit. Yeah, Rohit, can you say what?
So I'm looking at animal stress, just different type of stress during this day-to-day life. So just I'm looking at different what the effect of stress on animals.
What he defined is the question or the objectives, but you need to, when you make that kind of answers, you also need to say why that is important to study that, right? You may always say, these are my objectives. These are the question that I'm asking. But you need to say why this question is important. So that is called as defining the problem. If you see the slide, I said provide a brief introduction on the broad topic, define the research question, and state your hypothesis, and then say how are you going to test your hypothesis. That's what called an approach. And if you have results, and what do they mean, that's the conclusion that you do. So even if your answer is only 3 or 4 sentences, they should have all these things. Okay, so you have to remember that, and it's not easy. You have to really rehearse many times and then do that. So with this, we are ending our course, Introduction to Professional Scientific Communication. So I hope all of you had some understanding on how effectively you can communicate, whether it is writing or making a oral presentation, and even during discussion, because the last session, a kind of an interactive session, really focused on a kind of a discussion, right? So that I hope that it was enjoyable, and you all could benefit out of this particular course, and I wish you all the best in your research endeavor.